And this is we are discussing the ribosomes. Ribosome, it is simply the protein factories. Ribosomes are simply the protein factories. Here we are discussing initiation of protein synthesis. This protein synthesis, it occurs on the ribosomes, and these ribosomes are simply the protein factories. These ribosomes have got two subunits. This is small ribosomal subunit, this is large ribosomal subunit. Small ribosomal subunit and large ribosomal subunit, and this is the messenger RNA. This is messenger RNA, 5' to 3' end. This 5' end of the messenger RNA, this 3' end of the messenger RNA, and these are three sites. This is called A site, P site, and E site. A site is acceptor site, amino acyl acceptor site. P site is peptidyl site, and E site is exit site. In addition to this amino acid, it has a ribosome. It has a groove in a large ribosomal subunit, what is called as tunnel, or groove through which the polypeptide chain emerges after the completion of the protein synthesis. So we'll discuss about the initiation of protein synthesis. This protein synthesis, initiation of protein synthesis. How this protein synthesis is initiated. The smaller subunit of initiation of protein synthesis, the smaller subunit of a ribosome, it binds to the smaller subunit of ribosome, it binds to the messenger, it binds to the initiation factors, and this complex then binds to the messenger RNA. So we have got this smaller subunit of ribosome, it binds to the initiation factors. For example, we have initiation factor first, initiation factor second, and initiation factor third. It binds with the small ribosomal subunit. This is small ribosomal subunit. Initiation factor first, initiation factor second, initiation factor third. It binds to the small ribosomal subunit, and ultimately this complex binds to the messenger RNA. This 5' end, 3' end. This is messenger RNA. This is messenger RNA. And in this is messenger RNA. For example, we have AGG, AGG, AUG. How this initiation of protein synthesis takes place? The small ribosomal subunit, it binds to the initiation factors, and this complex in turn binds to the messenger RNA. So this protein synthesis starts while this there is a particular sequence that is called as Shine-Delgado sequence. Shine-Delgado sequence. This is Shine-Delgado sequence. AGG, AGG. This is AGG, G. This Shine-Delgado sequence, it precedes the initiation codon in case of prokaryotes. This is found in case of prokaryotes. And this initiation, this is Shine-Delgado sequence, AGG. It is the base sequence of 6 nucleotides. and it precedes the what? Initiation codon. This is initiation codon. This is what is Shine-Delgado sequence, and this Shine-Delgado sequence, it precedes the initiation codon, and this Shine-Delgado sequence, it is required for the efficient and effective translation of the mRNA. It is found in prokaryotes, while in case of eukaryotes we have Kozak sequence. We have Kozak sequence. This Kozak sequence, it is found around the initiation codon in eukaryotes. It is found in eukaryotes, and it has got a sequence of GCC, ACC, AUG, G. This is the Kozak sequence, and this Kozak sequence, it is found in eukaryotes, and it is found around the initiation codon. Both these Shine-Delgado sequences found in prokaryotes and Kozak sequence is found in eukaryotes. Both these sequences are required for both Shine-Delgado sequence and Kozak sequence are required for effective and efficient translation of the mRNA. Both Shine-Delgado and Kozak sequence required for efficient and accurate translation of mRNA. This Shine-Delgado sequence, it was discovered by these two scientists, Shine and Delgado, and Kozak sequence was discovered by Kozak. It was discovered by Kozak in 2002 in case of eukaryotes, and Shine-Delgado sequence is found in prokaryotes. We have got two initiation codons. This initiation of protein synthesis, I told you that during the initiation of protein synthesis, small ribosomal subunit, it joins with the initiation factors. These complex then binds to the messenger RNA. In the messenger RNA, around the initiation codons, we have two types of sequences in prokaryotes and in eukaryotes. In prokaryotes, we have got Shine-Delgado sequence. It is the base sequence of 6 ribonucleotides. We have AGG, and then in eukaryotes we have a Kozak sequence discovered by Kozak in 2002. Then what is this sequence? It is GCC, ACC, AUG. This sequence is found around the AUG codon, initiation codon in eukaryotes. Both these Kozak sequence and this Shine-Delgado sequence, they are required for the efficient and accurate translation of the mRNA. Then we have got two types of codons. We have two types of codons. One is called as initiation codons, and is called termination codons. We have initiation codons. First we will discuss initiation codons. Initiation codons are AUG and GUG. AUG and GUG. This AUG, it is an initiation codon. This AUG, it is an initiation codon. It codes for formyl methionine in case of prokaryotes, and AUG codes for methionine in eukaryotes. It codes for methionine in eukaryotes. Then we have the GUG. It codes for, basically it codes for two amino acids. One is methionine and then valine. It is an ambiguous codon. It codes for two amino acids. One is methionine and valine. If the GUG codon is present at the initiation position of the mRNA, then it codes for methionine. If this GUG codon is present somewhere other than the initiation position in mRNA, then it normally codes for valine. So we have two initiation codons, AUG and GUG. So this protein synthesis will take place like this. So we have what is messenger RNA. We have messenger RNA. It is 5' to 3' end. It has, these are codons. This is small subunit of ribosome. It means that small ribosomal subunit So protein synthesis will start like this, and this small ribosomal subunit, the mRNA, messenger RNA, it will get attached to the small ribosomal subunit. This is messenger RNA. Suppose it is AUG codon. This is 5' end, 3' end, small ribosomal subunit. It has two sites. One is A site, then we have got P site, then we have got E site. This is called as initiation complex. That means mRNA binds to small ribosomal subunit. Then what happens? The first tRNA will come. That means, for example, formyl methionine. First tRNA will come, formyl methionine. It comes to the ribosome at the P site. That means it would be like this. This is small subunit of ribosome. This is messenger RNA. Small subunit of ribosome. This is, for example, AUG. The first tRNA will come from the P site of the ribosome. It will come via the P site of the ribosome. So it would be like this. So for example, this is formyl methionine. Formyl methionine in case of prokaryotes and methionine in case of eukaryotes is the first initiator. This is initiator tRNA. It arrives. This initiator tRNA, it arrives at P site of ribosome. G, it is for G links with C. This is the anticodon of tRNA. G links with C, U links with A links with U. So the linkage between the anticodon and codon, this is codon of mRNA, is established. The linkage between the codon and codon of mRNA and anticodon of tRNA is established at the P site. because the first tRNA, it arrives at the P site of the ribosome. Then what? Then now the large subunit of the ribosome joins to this complex. So in the next step, large subunit of the ribosome joins to this complex. So we have got this. This 5' 3' and we have AUG, then UAC, this is tRNA, formyl methionine. Then the large subunit of ribosome binds to this complex. This is large subunit. That means large ribosomal subunit joins the complex. This is messenger RNA. This is initiation codon of messenger RNA. This is tRNA. This is anticodon of tRNA. This is the first tRNA, initiator Trna, which arrives at the P site of the ribosome. Then what happens? Now this is the initiation of polypeptide synthesis. Then we have got another is elongation. Another step is elongation. Elongation of polypeptide chain. Then we have another step, that is elongation of polypeptide chain. Now this tRNA, another tRNA will come to the ribosome from the A site. This is second tRNA arrives at A site. Remember that only one tRNA arrives, only the first tRNA arrives the ribosome from the P site. All other tRNAs, all other charged tRNAs, they arrive from the A site of the ribosome. That means they arrive from this site. So second tRNA arrives at A site. It would be like this. It would be like this. This is suppose messenger RNA, 5' to 3'. This is small subunit of ribosome, large subunit of ribosome. This is P site, A site. This is E site. We have AUG, then we have got UAC. This is formyl methionine. Then the second tRNA arrives at A site. This is second tRNA, amino acid second. So it will arrive at the A site. So it would be like this. Amino acid second will arrive at the A site. Now what happens? The covalent bond between the first tRNA, and its amino acid, that is broken, and then the peptide bond is formed between this amino acid and the second amino acid. So we can see that covalent bond between the covalent bond between first tRNA, the covalent bond between first tRNA and its amino acid is broken. Covalent bond between first tRNA and amino acid broken. A peptide bond is formed between the first amino acid and the second amino acid. Peptide bond formed between first amino acid and second amino acid. Obviously it would be like this, and the enzyme which is responsible for the formation of peptide bond, that is called as peptidyl transferase. Peptidyl transferase. This is a ribozyme. This peptidyl transferase, it is a ribozyme and it is present in large subunit of ribosome. It is present in ribosome. So it would be like this. Next step, what would happen? This is large subunit of ribosome. This is smaller subunit of ribosome. This is E site, P site, and A site. So we have got this tRNA at the P site. This is amino acid first, that is formyl methionine. This is amino acid second. Now the peptide bond is broken between this amino acid and this amino acid, that is formyl methionine amino acid and amino acid second. Yeah, we can say that formyl methionine, that is amino acid first, formyl methionine. So it would be like this. This bond is broken and then it will be here. That means a covalent bond is broken between the initiator tRNA and its amino acid, that is formyl methionine, and then the peptide bond is formed between the first amino acid and second amino acid. It would be like this. This is messenger RNA. It's 5', 3'. This is E, P, and A. Now the tRNA at the A site, the tRNA at the P site, it is without an amino acid, and the tRNA at the A site, it is having 2 amino acids. This is first amino acid formyl methionine and second amino acid second. Now translocation will take place. Now translocation. Translocation. What do you mean by translocation? Translocation means the sequential movement of tRNA from A site to P site and then to the E site, coupled with the movement of codons, and that is called as translocation. That means movement of what is translocation? Movement of tRNA from A site to P site and then to E site, coupled with movement of codons of mRNA. This translocation, it requires what? It requires the enzyme translocase and GTP. It requires enzyme translocase and GTP. So it would be like this. This is here. We have 5' to 3'. We have larger subunit of ribosome, smaller subunit of ribosome. We have PAE site. The tRNA at the P site. This tRNA at A site, it has 2 amino acids, formyl methionine amino acid and amino acid second. Now this tRNA at the P site, it is shifted to E site, and so we can, this tRNA which is present at the P site is shifted to E site, and tRNA which is having dipeptide at the A site, it is shifted to P site. So this is known as translocation. Translocation means that this tRNA will get shifted to exit site, and the tRNA present at the dipeptide which is shifted to P site. So it would be like this. It would be at the P site. This is known as translocation. Amino acid second, this is formyl methionine. So in translocation, what happens? There is the sequential movement of this tRNA from A site to P site, and from P site to E site. So I have shown it here like this. This tRNA, it shifts from A site to P site and E site. The first tRNA, this is initiator tRNA. The initiator tRNA, which is without, this is what? It is initiator tRNA. This initiator tRNA gets shifted from P site to E site. Then it slips away from the ribosome. Then we have got P site and A site. Now the second, third, we can say that next, this was the second amino acid just carried. Then the third amino acid will be carried by the tRNA from the A site. So this is next amino acid third. Next amino acid, that is amino acid third, will come from the A site. So it would be like this. It would be like this. We have messenger RNA, 5' end, 3' end. Then we have E site, P site, A site. At the P site we have 2 amino acids, formyl methionine and amino acid second. At A site, now next tRNA will come, and it will be at A site. This is next amino acid carrying tRNA. Now in the next step, what will happen? The covalent bond between this tRNA and this amino acid is broken, and the peptide bond is formed between this amino acid and this amino acid, and in this way the polypeptide chain grows. It would be like this. 3' 5'. We have P site, E site. We have P site and A site. So peptide bond is broken. This is amino acid third. So second amino acid, first amino acid. This is amino acid second. This is amino acid first. So this uncharged tRNA, that is tRNA without any amino acid, it moves to the E site and then slips away from the ribosome. This this tRNA having the 3 amino acids at the A site, it is being shifted from A site to P site. In this way the polypeptide chain grows. So it would be like this. We have P site, A site, E site. At the P site we have three amino acids, amino acid 3, amino acid second, this amino acid first, that is formyl methionine. Now fourth tRNA will come, amino acid fourth. In this way this polypeptide chain grows. So we can say that what happens in case of elongation of polypeptide chain, that means second tRNA arrives at the, this is first tRNA, the first tRNA will arrive to the ribosome from the P site. This is formyl methionine, that is first amino acid. This the anticodon of this tRNA will establish the linkage with the first codon, initiation codon of the mRNA. Then the next, that is the second tRNA will come, but it will come from the A site. Only the first tRNA comes from the P site, peptidyl transferase site, whereas all the other tRNAs, charged tRNA, they come from the A site, acceptor site, amino acid acceptor site or amino acid acceptor site. So this is second tRNA will come from the A site of the ribosome. Then the covalent bond between the first tRNA and its amino acid is broken, and the peptide bond is formed between the first amino acid and the second amino acid. This peptide bond is formed by the enzyme that's called as peptidyl transferase. This is the ribozyme which is present in the ribosome. Then we have got another important step that is called as translocation. Translocation means the sequential shifting of tRNA. It is the sequential shifting of tRNA from A site to P site and from P site to E site, followed by coupling with the movement of the codons of the mRNA. That means when this tRNA moves from A site to P site, then simultaneously the movement of codons which are present on the mRNA, that will take place. That means all the codons which are present on the mRNA, they reach to the A site of the ribosome and get decoded. That means the ribosome, this is for example mRNA, this is messenger RNA, This is messenger RNA, for example, this is messenger RNA, and the ribosome, this is ribosome. It physically moves along the mRNA. As a result of these, all the codons which are present in the mRNA, they get exposed at the A site. So we can say that this messenger RNA and ribosomes, we can say that the ribosome moves on the messenger RNA in the 5' to 3' direction. It physically moves on the messenger RNA in the 5' to 3' direction. So this ribosome, it moves on the mRNA in the 5' to 3' direction, so that all the codons which are present on the mRNA, they get exposed at the A site and ultimately get decoded and ultimately get linked with the anticodons of the tRNA. So for then next step is that this next tRNA will come at the A site, but the tRNA which is present at the P site, it gets shifted to the E site. Prior to that, there is a breaking of covalent bond between the tRNA and its amino acid. So this bond is broken. This tRNA will come at the A site. Then the bond is broken between this tRNA and its amino acid. This amino acid will shift it from this site to this. So peptide bond is formed between the amino acid third and second amino acid. This is done by the peptidyl transferase. In this way, polypeptide chain grows. And there are certain important factors that are called as elongation factors. Elongation factors, they are required for the elongation factors. They are required for the elongation process. Elongation factors, what are called as elongation factors, they are required for growing of the polypeptide chain. Another important thing is that for incorporation of each amino acid in a polypeptide chain, how many 1 ATP and 2 GTP molecules are required. That means in a polypeptide chain we have got many amino acids, and for the incorporation of each amino acid in a polypeptide chain, energy is required. And how much energy is required? 1 ATP and 2 GTP molecules are required. So 1 ATP and 2 GTP molecules are required for the incorporation of each amino acid in a polypeptide chain. This is what is called as this is what is known as elongation of polypeptide chain. Then we have got termination of polypeptide synthesis. Remember that this ribosome physically moves along the mRNA in the 5' to 3' direction. That is, ribosome physically moves in 5' to 3' direction on mRNA. And this ribosome, it physically moves in 5' to 3' direction on the mRNA. Then we have got termination of polypeptide chain. Termination of polypeptide chain. also remember that magnesium is required for the magnesium ion is required for the association. Magnesium ion required for association of ribosomal subunits. Association of ribosomal subunits. This magnesium ion is required for the association of ribosomal subunits. Then we have got termination of polypeptide chain. 3 termination codons. We have UAA, UAG, UGA codons. These are called as ochre, amber codon, and opal codon. These are known as termination codons or stop codons, or earlier they were called as nonsense codons. Actually, they are called as stop codons or termination codons. When this tRNA is not able to read these codons UA, UAG, UG, and this when they come on the, for example, this is mRNA. We have got AUG, UUU, UGG, AAA. We have, for example, UGA, UGA, or UAG, or UAA, UA, UAGG. These are not these 3 codons. UAA, UAG, UGA. These are not they are not read any tRNA. As a result of that, they signal the stop of polypeptide synthesis. Stop of polypeptide synthesis. They signal for the stop of polypeptide synthesis. They signal for the stop of polypeptide synthesis. And these ribosomal subunits, also there is decrease in magnesium concentration, and this causes the dissociation of the ribosomal subunits. In this way, there are also dissociation factors, which these dissociation factors are responsible for the dissociation of ribosomal subunits. Thanks.